All right, so he started the recording, and we'll start. Hey everyone, speaking with us today is Kaela Singleton, a postdoctoral researcher at Emory University and co-founder of Black In Neuro, who was also featured in Forbes 30 Under 30. Ms. Singleton, would you like to tell us a bit more about your background in research?
Yeah, I can definitely do that. So is this the part where you want me to share the slides? Sorry.
Yeah.
Yes. Okay. So I can definitely do that. And I have a couple of slides here that I'll walk you through to let you know more about like me as a person, but also the research that I do. So growing up, I grew up in Texas. My parents met in the Army, and they were super supportive of me as like a tiny human. They didn't really care like what I did in life. Their biggest thing was that I would go to college. And so growing up, I really only had like three rules that I had to live by. The first was don't add or subtract from the population, which I think makes a lot of sense. The second was if I was ever in doubt about if I should do something or not, to ask myself, will this upset your mother? And I still use that rule to this day. Like sometimes you do it anyway, but like you have to be cognizant. And then the third, like I said earlier, was really like go to college. I had to go to college. And I think that that's a universal theme that a lot of Black people, but also people of color in America, their families like want for them is to get an education. But since I didn't really have any like, I don't know, I didn't have to be a doctor or a lawyer. They just wanted me to go. So I had a lot of freedom in picking careers. And so my first career that I wanted as a kid started in 1992, right when I was born, and I wanted to be a Christmas elf. And I've always been a really big proponent of the holidays. I love Christmas. I love the holiday season and like winter, especially. This career goal, however, was ruined for me by a gentleman of the name of Clark Fisher in the third grade when he told me Santa wasn't real. And I've never forgotten him. Um, so after that first dream of being a Christmas elf, I had to pick a new job or a new career choice. And so I decided that I wanted to be a woman that had a nice office in the way that like women in romantic comedies and in a lot of TV shows, they don't really have like jobs. They just have really nice offices. And I was like, I could do that. I could have a nice office with a door that closes, and that would be great. And then in high school, I decided, or in high school, I got the opportunity, or middle school, high school, I got the opportunity to do this science research activity where we got to dissect all of these different sheep brains. And that's how I discovered that I loved neuroscience. And that's what I wanted to do. So at the ripe old age of 12, I decided that I would study the brain, and I picked where I wanted to go to college from there, like what I wanted to study in college from there. And it informed my decision of like what I would do. So really, I was like super stubborn. And then after college, I decided that I wanted to go to graduate school. And most of my goals after that were to just make it through graduate school because it was a really hard time. When I think about my career goal now, though, and all that I've accomplished, it really is like encapsulated by this Maya Angelou quote that focuses on this idea that life is not simply just for you to survive it, but for you to thrive and to do so with passion and compassion and style and humor. And I feel like I've done pretty good at that this thus far. So now I'm a postdoctoral fellow at Emory. I'm an adjunct professor at my alma mater, Agnes Scott College. Um, I was featured recently in Forbes 30 Under 30. I'm a D-SPAN scholar, and I've won tons of awards. And so much of what I do is because of the unconditional support from my family, but also because of my passion and love for neuroscience, specifically my love for neural development and being a developmental neuroscientist. And one of the reasons that I love neural development so much is because I think it's really similar to how we train and build scientists. And so I'm going to walk you through that sort of parallel, right? So at the very beginning of neural development, the foundation of it that happens across like all organisms, you start off with these common progenitor cells that over time divide in order to increase the surface area of the embryo. These common progenitor cells will eventually go on to be neuronal progenitors where they'll exit the cell cycle and begin expressing increases in proneural genes. And now their job is really just to make the cells of the brain. Over time, these neuronal progenitors will become immature neurons. So neurons that are really like trying to figure out where they want to go, the connections that they'll make, they're not really fully formed and they don't really know what they're doing. But eventually they'll maturate into the mature neurons that we're used to seeing, right? It'll be neurons that have specific functions, neurons in your eyes and your ears versus that in your prefrontal cortex, and they'll look different. And what controls this process over time, or what orchestrates it, is this balance that has to be maintained between these sort of extrinsic regulators, things like environmental factors, chemokines and chemoattractants, so like literal toxins and signals, and these intrinsic factors, things like DNA and RNA and downstream targets. And that has to be tightly maintained and controlled in order for us to get the sort of the diversity that we see within the brain. Excuse me. And what I think is really interesting is that this is really similar to how we develop scientists and how people's scientific interests in like evolve over time. And that's all scientists, right? Not just neuroscientists, but for the sake of this, that'll be the example that I use, where you start off when you're interested in science with many scientific ideas, right? You have to take classes in everything from plant biology to cell biology, maybe some organic chemistry in there. And over time, if your goal is to be a neuroscientist, you'll exit this general science studies and you'll begin expressing this increase in neuroscience overall. Eventually, you'll become a student or a trainee, right? Where you're learning new skills, you're trying to figure out what type of research you want to do long term, you're being mentored by someone and they're helping you figure out what you want to do next. And eventually you'll maturate into becoming a mentor, right? Where you'll help other people figure out what their goals are and what type of science they enjoy doing. And so for me, this process obviously is really similar to how the brain naturally forms. And when you think about it, the same thing that controls this, that controls neural formation, also controls this, right? If we take me for example, the things that are really important in my development as a scientist are these intrinsic factors, right? My identity as a Black Samoan queer woman growing up in the South. And the extrinsic factors represent like my choices, right? Where I decided to go to college, the fact that I went to an all-women's college or an interdisciplinary program for graduate school, or the funding that I earned throughout that time. And then another big extrinsic factor are your friends and your mentors, right? Everyone's from the PIs whose labs you work in to the friends and peers that you make throughout this process. I think another really big extrinsic factor for me is Black in Neuro and building that nonprofit and community with my friends, where each week that we've done Black in Neuro Week, which really our goal is just to increase the visibility and community of Black neuroscientists or people interested in neuroscience, has been life-changing and life-altering. We've had this sort of global reach that's allowed us to connect with so many people, and that directly influences my decision to stay in science as a whole and to continue to pursue it and the type of research that I study. And I think one of the reasons that I love this metaphor and this kind of work is because it goes really well with sort of the overall branch of the research that I do now, which is under the umbrella of systems biology, which I like to describe as a puzzler's dream. It's this, as the definition is on the screen, but it's an approach really that emphasizes looking at the bigger picture of what's going on and combining all of the sort of fine details that you get from studying things like genes and molecular networks to things like cellular networks and organs that can help us individualize patient treatment and care, but also can tell us a lot about the social dynamics of different disease states and how it looks different across things like class or race or ethnicity as well. Excuse me. And so the disease that we study in my lab that I work in currently, the Fonda's lab, is Menkes disease, which is a rare neurodegenerative and neurodevelopmental disorder. The symptoms of Menkes disease vary a little bit, but a lot of the physical symptoms that we see in patients is kinky hair and sort of pigment dilution, so being really pale-skinned. We see a lack of collagen, as well as things like cell death and muscle weakness, and things like intellectual disability, as well as epilepsy. These here are two just cartoon schematics of data that we've gotten before, where A shows us sort of the decrease in white matter within the brain, and C shows us the degeneration that I was talking about, where you have these normal neurons here called wild type that are really vibrant and they have all of these branches, but in our Menkes patients, they're sort of like shriveled and sad. I feel like that image really speaks for itself. But one of, I think, the most devastating things about Menkes disease is it affects most neonates, and most of the patients don't live past the age of 5. And so a lot of the questions that we serve to ask is, how does Menkes disease work and the mechanisms behind it, and how does it help us address bigger questions in neurodevelopment altogether. And so we know Menkes disease is associated with a deficit in copper in the brain. And so this is a really, really busy schematic. And all I want you to really gather from it is that Menkes disease is really important for development. It's important for things like mitochondria, integrity and localization. It's important for cellular trafficking. And it's just important for the synthesis of like compounds in the brain overall. And so our work really tries to understand how copper plays a role during development, since disruptions in copper have been associated with tons of neurodegenerative disorders, things like Alzheimer's and Parkinson's, but also copper-specific disorders like Menkes and Wilson's. And so I use fruit fly as one of the models to do the research that I do. And a big part of that research is trying to sort of recapitulate or make those Menkes phenotypes that we see in patients occur in flies. And to do that, we use this GAL4 driver system where we'll cross two flies using basic Mendelian genetics to give flies our sort of Menkes phenotype so that we can ask questions that we aren't able to ask in human patients. And when we do this, right, when we take these flies and we give them this Menkes phenotype, what we see is that they have this deficit or decrease in synapse morphology and synapse formation. And by doing different genetic mutations, we can rescue this on some level. So these are fly neuromuscular junctions that have been stained in green with HRP. And what hopefully you can appreciate is that in our control compared to our Menkes, these first two panels, we see this reduction overall in synapse length, and that's quantified for you over here. And when we look at a mutation in COG1, which is a molecule important for trafficking, we're able to rescue on some level this phenotype here in this last row. And so this led us to ask a lot of questions about the morphological challenges and synapse challenges occurring in these flies. Is this similar to what we see in patients? And the answer is we don't know yet, but we were able to do more investigations using this mechanism. One of the things that we did find that is really interesting, which is what my project focuses on, is looking at mitochondrial content. And if you'll remember from like school, mitochondria is the powerhouse of the cell, but it's also really important for other functions like homeostasis, so making sure that cells have enough energy, that they're alive, that they can communicate with one another, but also the idea that mitochondria are mobile. And so what we did was we used this specific labeler along with our Menkes overexpression phenotype to try and figure out where the mitochondria are within the cells of these flies, right? So the first is our control here, where the neuroplasma membrane is in like magenta and the mitochondria are in yellow. And when we look at our diseased flies, right, our flies that have our Menkes phenotype, we see that there are very little mitochondria within the synapse. But when we look in our rescue flies, we see that we're able to fix that, right? So what I think is most interesting and what most people ask when they see this data is, well, where are the mitochondria in these animals? If they're not in the synapse like they are in all of these other categories, where are they? Where could they possibly be? And interestingly enough, the mitochondria appear to be stuck in the soma of these flies. So these are images taken of the cell bodies of the neurons that I just showed you. But hopefully what you can appreciate here is all of this yellow here is mitochondria within the cell bodies of those Menkes phenotypes. And so when we think about this, the best way that I like to think about it is where the mitochondria, this idea that they're stuck in traffic. And I hate traffic. I live in Atlanta, and I don't think traffic is as bad as it is in California, but where I live, traffic is still pretty horrible. But the way that I like to think about this is that all of those mitochondria that are stuck in the cell bodies is equivalent to a bunch of cars being stuck on the highway. This is like a real picture of Atlanta traffic that was like taken a couple of days ago. Anyway, it's a bunch of cars stuck on the highway. And so there are two reasons for this kind of traffic, right? The first is that there's literal damage to the road, right? That there's some sort of like catastrophic thing that's preventing the cars from driving up and down the highway. This is a real picture of a highway bridge collapse that happened a couple of years ago in Atlanta. The cellular equivalent of this is there damage to what shuttles mitochondria up and down like through the cell in and of itself. So is there damage to the motor proteins that help mitochondria move? The second option is that there are too many accidents, right? In Georgia, we have this thing, it's called I think it's called Hero 511. You can call them if you get into an accident and they'll come help you, like leave your car and call the police and stuff like that. But you can imagine if there are 100 accidents on the road, there's no way that they're going to be able to help anybody. So it's just going to cause a bunch of traffic and there's going to be a bunch of traffic jams. The cellular equivalent to this is really thinking about this as is there a way, are we damaging how damaged, are we ruining how damaged mitochondria are cleared, or are we messing with something called mitophagy, which is how damaged mitochondria are removed from the cell? And so my project is really focused on looking at option 2, this idea that there are too many damaged mitochondria in the cell and not enough sort of degradation mechanisms to get them out. And when we do this experiment, where we take our ATP7A overexpression, right, our Menkes phenotypes, our Menkes flies, and then we cross them with different players in the mitophagy pathway, what we see is that we're able to rescue that. We're able to fix the synapse morphology. And our hope is that we're also able to rescue mitochondrial localization, but we haven't done that yet. One of the things that I do want to explain, though, because I feel like this is a little counterintuitive, is that the idea here is that damaged mitochondria recruit both pink and parkin, right? When a mitochondria is damaged, they call both of these molecules over, and then that damaged mitochondria is taken up by the lysosome in the cell. It's removed and like degraded. And so what we're saying here is by preventing this from happening, we're allowing more damaged mitochondria to move throughout the cell. And that in and of itself doesn't necessarily make sense. So we're still trying to piece together how it is that having damaged mitochondria is better than having no mitochondria overall. And so that's like a big picture idea of some of the work that I do, where we're trying to really piece together this story of how pink and parkin play a role in copper damage within the synapse, but also what role mitochondria play overall in neurodevelopmental disorders and in neurodevelopment as a whole. And this work is funded by my own grant, but also grants to our lab and all of our collaborators. And yeah, so I'm happy to answer any questions or go over anything. I feel like I kind of ran through that, but happy to keep chatting.
Sorry. Hi, Ms. Singleton. Thank you so much for presenting. With that, we can move on to our Q&A. So for our first question, what was your inspiration behind creating Black for Neuro?
Yeah. So a big part of our inspiration for making Black and Neuro was in the early phase of the pandemic. There was a lot of social injustice and social unrest, and one of the biggest sort of like consequences to that was seeing how it affected like other Black colleagues at our institution and in like at my institution and in other institutions. And so it really actually started with a tweet from one of my friends asking if we were going to have a Black and Neuro week where we highlighted the work done by Black scholars. And since then, there have been tons of other Black and X weeks. There have also been some Latin and Chem and Latin and Neuro weeks as well. And so it really was just a need, I think like a social need for people to have a better sense of community and like find people that they could talk to.
So I have the next question here. So what motivated you to study Menkes disease in particular?
Yeah, so one of the things that I find really interesting about Menkes disease and most neurodevelopmental diseases is that we actually like know the cause. We know what mutation is responsible for the disease, but we don't necessarily know how to fix it. And there's just a lot of research that can be done and needs to be done about just sort of basic cellular biology, right? Like how does copper affect cells? What is copper important for? And I should mention too, I had no idea that copper was important for like brain development, despite having like a literal PhD until I joined the lab. So it was also just really cool to me. Like it was intriguing. I knew nothing about it.
Okay, so shifting from your research, for our next question, we just want to ask about about how you got into fellowships and what advice you would give to aspiring scientists to get accepted into other fellowship programs like yourself?
Yeah. So I think I got my first fellowship when I was in college. I feel like now y'all are like way smarter and way cooler than I was at your age though. So there are a lot of like high school fellowships or like fellowships available for high school students. I didn't have that and I didn't know about it, honestly. But I think one of the biggest things that like really sets fellowship applications apart is like talking to the people writing you letter of recommendations and like telling them why you want this fellowship so that they can like personalize that letter for you. I think that plays a big role. I also think that there's like some skill and finesse in writing personal statements, and getting a lot of feedback on that portion of your application is really important because your personal statement is kind of like the big thing that everybody looks at. And once you once you nail a personal statement, like once you get one fellowship, it's easy to tweak your personal statement from there on out to like fit it to other ones. But it's really like having those letter of recommendation writers write a personalized letter, and then also having people edit your personal statement so it comes across really good.
Okay, so here's our final question. How can high school students get involved with neuroscience research?
Yeah. So I think there are tons of ways. And I didn't realize this until I actually was in graduate school because we had some high school students come work in our lab over the summer. One big way is just like Googling the colleges in your area and seeing if they have like they'll all have like biology or chemistry departments and things like that, but seeing if they have a neuroscience department and just like sending emails to faculty members. I know it can seem like really scary, but we genuinely get so excited when like we get emails from high school students, because we get emails from like other adults who like want difficult things from us, but like giving someone the opportunity to shadow in a lab is like really great. So one way is just like cold emailing people. I think another really good way is Googling to see if there are any fellowships in your area and talking to your science teachers like in school. A lot of the times they'll know about opportunities and programs. But I think those are the two biggest ways. And then just like I don't know, however y'all found me, just like Googling on the internet, like asking people if they know. And I promise like 9 times out of 10 people are going to be so excited and enthused to help you, or at least like put you in contact with like the right people.
Okay, that's great. Well, everyone, thank you so much for watching our interview with Miss Singleton. Take care, and we'll see you all next time. Bye.
Bye.